I think we can start the discussion, and we have, let me introduce first Laura Andres Martin, who is a research investigator in oncology at the New York Stem Cell Foundation Research Institute. She's leading the ovarian cancer precision medicine program. And she's an expert in cancer signaling pathways and adult stem cell biology. And she's developing stem cell-based technologies to generate patient-specific models for cancer research, including the generation of multiple organoids from ovarian cancer research. And she's going to be our fourth panelist. And with that, I think that we can start a discussion on the future of cancer therapies. But before, we have a question in the chat for Sohrab. So have you looked for the antigen presentation profiles across the ovarian cancer tissue types? Any insights there?
So if I understand the question, so certainly there is variation in HLA expression, the antigen presentation machinery, and this is highly expressed in the HRD tumors, but not in the HRP tumors. So there's definitely a association with the mutational processes in the expression, is cell intrinsic expression of antigen presentation machinery as well. I didn't go into that, but that's certainly part of the cell. There are cell intrinsic malignant cell properties that also co-associate with these mutational processes.
And I mean, I understand that your work is primarily on high-grade serous ovarian cancer, which is about 70% of advanced ovarian cancers. But I've been wondering for a long time whether high-grade serous is really the most immunogenic ovarian cancer, or we would be better focusing on clear cell or mucinous or other tumors that.
Sure, yeah, absolutely. I mean, I think clear cells are certainly have APOBEC mutational processes that in about half of the cases, and then in the endometrioid setting we have MSI, and so those would certainly be great candidates for immunotherapeutic approaches. But I think our work is very much focused on trying to dissect the variation within high-grade serous ovarian cancer, and even that, I think we're really finding that these are histologically similar disease, but there are really definable patterns that can substratify the cases and probably merit different treatment modalities on top of PARP inhibition that's already been established.
There is another question for Padmanee. Have you applied the EZH2 inhibition approach to ovarian cancer, which also has a very low tumor burden?
No, we haven't. So we just opened the trial, and we're doing in the dose escalation phase. As you can imagine, we need a small dose escalation, and then we'll have the dose expansion for that combination, since this is the first combination of an EZH2 inhibitor and an immune checkpoint therapy agent. So no, we don't have it in ovarian yet, but if we see good data, because we're getting pre and post treatment samples, so we'll have a better understanding of how the tumor microenvironment is changing to propose additional studies.
Okay. I mean, ovarian cancer, I think, is a fascinating disease to me. I don't know what you think, because it's a disease in which everybody agrees T-cell infiltration determines the outcome of patients, right? I personally think that the humoral response is as important, if not more, than the T-cell response in this particular disease, but T-cell matters. But immune checkpoint inhibition has not worked. Now one can say, yeah, it has low mutational burden, but renal cancer is right there, and immune checkpoint inhibitors work, right? So I think it's an example of a disease for which we need much better biological understanding, because I don't think that we really understand what determines the immunogenicity of ovarian cancer. Probably not neoantigens, but maybe gene fusions are more important driver of neoantigens that point mutations, right? Unlike melanoma, for
Yeah,
instance.
I mean, I think given the profound structural variations that exist, what hasn't been adequately explored is the expression of, for example, endogenous retroviral sequences in the genome and how those get affected by structural variations. I think that's a really fascinating question to explore. And I think what our data shows is that even in the disease natural history, the immune evasion mechanisms are really profoundly distinct according to the very etiology of disease, which is in the mutational processes. So I think we have to account for that when thinking about these next steps going forward when we think about therapeutic strategies.
Yeah, we have to think about how the tumor microenvironment has evolved over time to have different immune cell subsets and how those subsets can differentiate, right? I mean, if they're receiving certain signals, they will differentiate through a different pathway than if they're receiving, interferon signals. So it's I think we have to stop thinking of it as one size fits all, right? I mean, we really have to get back to basics. So yes, immune checkpoint therapy, when by that we mean anti-CTLA-4 and anti-PD-1, PD-L1, because that's really what we're talking about predominantly. So those are blocking inhibitory pathways on T cells, but we know that the immune response is controlled by so many other pathways, not just those. And when and where those pathways get expressed, and through which mechanisms, these are still unknown, and we have a lot more work to do because we're just at the tip of the iceberg, in my opinion.
Mm-hmm. Yeah. I just want to add to that in addition to that, the high heterogeneous nature of ovarian cancer, right? Whether different metastases may have, different tumor immune cell interactions, and how can we actually overcome that problem? We may study only one piece of tumor from this resection, but then how that applies overall to the whole spectrum of tumors that the patient presents, right? So I think that's another feature of ovarian cancer, very unique, or not so unique, but like one of the most predominant features of this disease that is very difficult to understand and to overcome.
So in that regard, how do you see the use of the organoids, for instance, that you are developing as a tool to characterize the heterogeneity of the disease and evolution?
Yeah, that's one of the things that I think we can achieve perhaps with modeling utilizing the organoid technology. So first we can derive organoids and grow from different metastatic sites or from different resections for the same patient. So we could envision to really have reproduce in the lab and grow in the lab the whole spectrum of tumors that the patient is presenting. And that's an advantage as opposed to, other tumor models that we have. PDXs, of course, is another high level of modeling tumors, but because of the difficulty of maintaining and, they are more of a harder to develop in the lab. However, organoids is something that you can grow in the dish. So it makes it more approachable in a way. And we could think of not only study the biology of that specific tumor resection for the same patient, but also we can, think of the functional testing, which is something that is becoming more and more of an application of the organoid technology on the different tumor sites and see how the responses goes for across different metastases.
And how can we overcome, in the opinion of everyone, the heterogeneity of the disease, right? If metastasis A has a different antigenic profile compared to metastasis B, I mean, how can we overcome that really? Obviously with a polyclonal response, but there could be common drivers. How do you envision that?
I think one of the things that we have to be thoughtful about, right, is that if we are looking at different metastases, we have to keep in mind, are they able to generate an effector T cell response? Because at the end of the day, it's an effector T cell. And I agree with you, B cells are important, as we showed in the tertiary lymphoid structure, right? You need T cell, B cell, dendritic cell, but can you really have those cells traffic and proliferate and survive within that tumor microenvironment, regardless of whether it's antigen X or antigen Y being expressed, right? How do those cells differentiate within that tumor microenvironment? And, again, as much as we would love to model things, the best model is always the patient. Because even, in a lot of these model systems, unfortunately, they're not immunocompetent. They don't give you that longitudinal basis of how to study an immune response dynamically. And that's been the issue in terms of trying to figure out how to do rational combination immunotherapies. So I think we have to really start thinking about smaller clinical trials, getting as much as we can from the patients and samples, studying those in real times, and applying some big data analysis, bioinformatics, computational biology behind it, so that we can start to really come up with what are the cell-cell interactions and how are they evolving within a real immune response, because the immune response is dynamic. All these resistance mechanisms we're talking about, it's not just about the tumors. It's the fact that the immune response intrinsically has these pathways to control itself, and we just don't know when and where they're being expressed.
Yeah, I would definitely support that. And I think our study that I presented today is studying tissues at diagnosis under primary debulking surgeries. But that's always also probably decades in the making, maybe 30 years of the evolutionary arms race between cancer cells and the immune response. And we're seeing the manifestation of that at a symptomatic time point. But there have really been many, many years of evolution that have gone on before we get to that state. And so the model systems will never be able to recapitulate all of the selective pressures that the host immunity is exerting on those cells and the mechanisms that the cancer cells actually have to then execute to escape immune surveillance. And so I think it's quite fascinating to study patients. Of course, we can do, much more interesting interventions and manipulations and model systems, and that's how we get at mechanism. But studying patients, I agree, is extremely valuable in unraveling these mechanisms of resistance.
Yeah, I think one component that sometimes we miss is how restrictive the tumor microenvironment is in terms of metabolic alterations, and how difficult it is for the poor T cell to exert its effector activity, because we worked with Juan Cubillos and Lori Glimcher a few years ago on a story on how you get activated T cells, you expose them to acidic fluid from ovarian cancer patients and they get frozen, right? And that happens through ER stress. So I think that some of these mechanisms could be targeted as adjuvants to support immunotherapy. And we need to work much more on that. You mentioned, Pam, the role of tertiary lymphoid structures, which is fascinating to me. We have several lines of research. I think that this is a new area, that has triggered the interest of the field, because as you mentioned, right, in different histological types of cancer, the presence of these structures were associated with better outcome and or better response to immune checkpoint inhibitors. And for those who are not immunologists or follow the field with a lot of detail, tertiary lymphoid structures are conglomerates of T cells, B cells, dendritic cells, follicular helper T cells that recapitulate the architecture of secondary lymph nodes, but they happen inside tumors. And what is fascinating for me about these structures is that we don't know how they are beneficial. Probably they are illustrating superior immunogenicity. That's obvious, because in order to orchestrate such a chunk of immune cells, you need some strong immunogenicity somewhere in the tumor. But I think the assumption is that the T cells there find a cozy home for under the influence of B cells that could act as antigen-presenting cells. And I am sure that this is absolutely true, but there is another factor, which is that there are germinal centers in this structure, so there is antibody production, right? And I think the antibody response is, it has been demonized for years, but in my opinion, that's based on models that don't really recapitulate the human disease in terms of the phenotype of the B cells and the isotypes of the antibodies that they produce. most of the antibodies that we see in mouse models are IgM. That's not what we see in advanced human cancer. We see IgG or IgA. So I think we need a lot to understand, because if really tertiary lymphoid structures are not only markers, but also they are doing something active to promote anti-tumor immunity, there may be ways of promoting them at tumor beds. So
absolutely. I think the tertiary lymphoid structures also tell us a lot about how we really need to start studying the immune response, right? I think we're so used to, you get your cells, you make your single cell suspension, and you do flow cytometry, or you do single cell RNA-seq, or you do nanostring, and you go, oh, I have an interferon signature, or oh, I have a CD8 T cell that's, interferon gamma positive. I should be seeing an anti-tumor response. And what the tertiary lymphoid structure reminds us, it's that you can have a high interferon gamma signature or lots of T cells and still see no anti-tumor response with an immune checkpoint therapy agent. And that's been a question mark for us a lot of times, because we're like, wait a minute, we have high TMB and high interferon gamma and still no response to immune checkpoint therapy. And what the tertiary lymphoid structure reminds us, it's that it's not that you have cells scattered, right? It's not that you can count them in a single cell data set. It's that they actually have to communicate with each other. And the distance between them matters, because that's how they're now communicating to help with pro-survival signals and pro-differentiation signals, so that you can get these kinds of memory responses. So I think, so that is one very important piece that we're learning, that, maybe some of our technologies gives us one set of information, but we really need to come back to what is the cell-cell communication. I think the other piece that it's teaching us is that we had all in our heads thought everything is happening in the draining lymph node, and the cells are being educated in the draining lymph nodes and then coming back into the tumor. I mean, it's, as an immunologist, that's the training I got from Immunology 101, right? I think what this is teaching us is that, maybe these cells actually are getting educated right in that same microenvironment. And you don't really need them to go into the draining lymph node, that they can indeed set the whole system back up within the tumor. And then the third and most important thing I think we're finally coming to terms with is that immune checkpoint therapy has this anti-tumor response, but it has these significant toxicities with them as well, right? And what we're starting to see, it's the same cell subsets that give you the anti-tumor response that gives you the toxicity. Every study we do, it's a CD8 T cell that gives you the anti-tumor response. It's a CD8 T cell that's expanding with toxicity. Now we're beginning to see, even in organs that, you're seeing tumor rejection in bladder and prostate, like we're saying with these clinical trials we've conducted, they're tertiary lymphoid structures. But now we're starting to see that in tissues that are being damaged with, nephritis, for example, we're now starting to see tertiary lymphoid structures define the immune-related adverse event as well. So it's really giving us a lot of information as we're studying this. And yes, I still think we have a lot more work to do to understand the B cell component and the antibody component. But I think it's a new realm for us to start digging into.
Yeah, totally agree. Yeah, because actually in autoimmunity, as you say, we see the other side of the TLS, right? It's a negative factor.
So Pam and maybe Laura, are there good models for recapitulating these tertiary lymphoid structures? Can we get there? Is it?
Yeah.
How difficult is it? And
so our postdoc's been working. I have a postdoc working on this question right now. Since we made these, we published the papers a few years ago now with Genwargo and melanoma, and then we published again with bladder cancer showing this. So, we have really nice autoimmune models, right? Because I think in the autoimmune disease space, they've noticed tertiary lymphoid structures associated with these autoimmune events. And so they have these models that you can look at that, you can try to propagate. Now, what we have to do is learn from the autoimmune disease setting and take that back into the cancer immunotherapy setting. So we have to go back and forth. And I think that's not something new. People have been doing that for a while. But I think that's where we're starting from, again, is in the autoimmune disease setting, to understand how these structures form and why they form, and the mechanistic understanding. And then maybe go back to a model where we can knock in or knock out a pathway to see if then you can alter or reinvigorate that kind of a setting in a cancer model. We're not there in a cancer model yet, but I think it's still early days, like I said, for studying that.
Mm-hmm. Yeah, I think I should make a trip to your lab and then bringing that back for the ovarian cancer. Well, maybe not modeling that specifically, but for immune cell interactions, or rather maybe testing some of these, new technologies, cell therapies. There is like this, let's say, next generation tumor modeling organoids that has started with incorporating one by one as of now. So that is still a limitation, putting back some of these components of the tumor microenvironment, T cells, B cells, fibroblasts, so that we can recompose what really the tumor is, because the tumor is not only the cancer cells, as we all know. We heard earlier from some of the sessions, this vascularization, for instance, that the laboratory of Sahin Rafi has at Weill Cornell, where endothelial cells can recreate, and really like the tumor educating these vessels that are created in vitro. So that's something that we are trying now to do, to really bring this next level of innovation into the what we do, which is growing the tumors as a form of organoid. So hopefully that will get developed further, and we can understand a little bit more, or maybe trying to prioritize all these like potential combinations that we can imagine through the information we get from the patients, and see if we can like filter further things that can be actually bring into the clinic and being tested, because there is a limitation of what we can do in clinical trials, right? Because there is no time, not enough patients, not enough of anything. So maybe prioritizing these strategies in clinical trials can be something that we can do preclinically with some of these more innovations and way to model tumors. So hopefully more coming up in the next few years.
But I agree that this modeling is very important. It's because, I mean, when you think about how secondary lymph nodes are orchestrated, it's different, because you get LTI cells that are not present, as far as we know, in tumors, right, that produce lymphotoxins. So there should be alternative mechanisms. And I think the field has put the emphasis on fibroblasts being activated and producing CCL21 and starting creating this chemokine milieu that is conducive to the orchestration of all the pieces. We have a paper under review. I don't know what will happen to it, but my personal view is that the seminal element in the formation of tertiary lymphoid structure is something that could be obvious for immunologists, but it's not that obvious for whatever reason in the subfield, which is the conversion of a CD4 T cell into a follicular T helper cell, because TFA cells have the capacity to produce CXCL13 that attracts B cells, IL-21 that activates B cells. We found that it produced the cytokine light, which is a member of the TNF alpha family that activates fibroblasts and high endothelial venules to produce CCL21. We think that this is the cornerstone of the orchestration of TLS. There is also something about the peritoneal cavity that we don't understand at all, that we confirmed, because this was first published by Vic Engelhardt. And if you put the same tumor in the peritoneal cavity, it develops TLS spontaneously. If you put it in the flank, it does not. Go figure, I mean, one could think that, they are sucking up the cells from the spleen, maybe the B cells. I don't know. Yeah. I don't know.
I didn't really go into this, but part of our approach has been to do a multi-site investigation of these tumors, and so what we see anecdotally is certainly the organ site itself has a kind of a soil effect, where you have a seed and soil effect, and certainly we see really quite dramatically different immune cell phenotypes in certain organ sites versus others, and of course that's the intrinsic nature of where the cancer cells are landing will pattern how the immune response and the tumor microenvironment properties, and that I think is something that you mentioned this earlier, Laura, but it has to be a factor in first of all creating variation, and secondarily then when a selective pressure is exerted on that variation, there'll be differential response.
I would like to tackle an issue before we move to cell therapies, which is close to my heart. So Pam, I mean, how do you think we can, further optimize immune checkpoint inhibition, right? I mean, I am sure we are not plateauing. You have pioneered studies, right, on how there are compensatory pathways that could be targeted. So what are your thoughts on that?
So some of the things, again, I think we have to be very thoughtful in what is it that we want to target in which patient setting. So are we targeting in neoadjuvant setting? Are we targeting in metastatic disease setting? Are we targeting after chemotherapy treatment, before chemotherapy treatment? All of these things make a difference in what we're talking about and what you want to target for the immune response. Are you targeting when it's a mostly myeloid-rich tumor? Are you targeting when it's mostly a T-cell-rich tumor? All of these things, I think, matter. And we're seeing now more and more myeloid targeting strategies. So anti-VISTA antibodies are out there, for example, TGF-beta inhibitors, as I talked about, is another one, epigenetic agents that can convert M2 to M1s. And these may all play very important roles in ovarian cancer, because I see that, they have different pathways when you look at their immune signatures compared to when you look at some of the other tumors like melanoma, for example. So I think these are some of the very comparative and simple studies. I don't think they need to be extensive. I mean, again, we do 10 to 20 patient studies. They're not large, but at least they try to answer one specific question at a time. And make some really comparisons with other tumor types that we may think of as more, the hot tumors or the immune checkpoint therapy responsive. And then the other thing to think about too is when we have the patient data, that's a hypothesis generating moment, right? You can't really test the hypothesis in a clinical trial at all. You're just generating the hypothesis. And the hypothesis then should be about scientific mechanisms that we think may be relevant. And maybe there's more than one, I don't know, but the clinical trial data has to be looked at very carefully, not just about what's the overall response rate and what's the survival data. I mean, that's great, and that's what we need. But even when we have a failure, we need to understand, why did we have a failure? What were the mechanisms that led to the failure? And especially when we have a success, what were those mechanisms? I think more detailed analysis to generate a hypothesis and then take it back in a way that you can really test it in an appropriate model, right? And then that gives you the data you need to come back with what might be the combination strategy, because I don't think in any tumor type we're going to get far without a combination strategy. You don't want to play whack-a-mole, right? You hit this one pathway and another pathway is the one that's now controlling the immune response. You want to understand that ahead of time so that you can come in with the right combination. So at least that's how I think about it, as we're planning studies. And I think that would be meaningful also for the setting of ovarian cancer.
Totally agree. Yeah. So I would like to, before we close, I would like to tackle your thoughts on cell therapies, because cellular therapies, CAR T cells, for instance, have revolutionized the management of hematological malignancies, but so far they have failed kind of miserably against solid tumors for a variety of reasons. I am still a believer that this is due to technical issues that could be overcome. And there is a lot of industry and academic interest in that. So, here at Moffitt, there is, cell therapy is kind of the driver of the immuno-oncology program. So I would like to get your thoughts from different perspectives.
So maybe I'll start and I'll say again, I think part of it is that in liquid tumors, you're dealing with a different tumor microenvironment and a different cell-cell interaction and signaling pathways, that solid tumors have multiple ways of overcoming, an immune response. I mean, the cell-cell signaling is a lot more because there are so many different other cells participating in this network. And the microenvironment, whether it's pH or oxygen levels, all of these are very different, and all of those things are going to contribute to how a T cell survives and function. So you may give, hundreds of millions of CAR T cells, And then they have to get into that microenvironment, and then they have to now survive and proliferate. And this is where I think understanding, again, the mechanisms that are at play will be helpful, because then you can engineer that CAR T cell to overcome whatever that specific mechanism is, right? Whether it's, it's now dealing with the hypoxic conditions, or whether it's now dealing with, IL-2 generating regulatory T cells that are suppressing. You have to come up with what is the mechanism maybe you can engineer out in that CAR T cell. And I think that, again, will come into more personalized treatment for the CAR T cell space.
Mm-hmm. Yeah, I would like to add to that, coming back to this combinatorial strategy, right? Like maybe something that we need to think about is how to perhaps target understanding the mechanism and then find some sort of small molecules probably, or something else that can, inhibit that inhibition of the CAR Ts entering into the tumor, and then take that combinatorial strategy into that specific patient, or, or in clinical trials. We can also think about leveraging, even like immunize the tumor in a way with also like some sort of drugs that can like trigger an immune response and leverage that side of, or that strategy would be another thing that would be interesting. And many things are putting an effort in that at the moment. Or there is other immune cells that could be using for, in addition to CAR Ts, we know, Callium or disciples from Callium works, and macrophages, NK cells, and how can we develop that further to putting all of them together maybe into better and more effective treatment. So all that is definitely ongoing, and more to come in the next years for sure.
Yeah, I mean, my personal view is that, I mean, this is all absolutely true, but I think the field can solve I mean, we can create super T cells through CRISPR genetic engineering, that don't undergo, XBB1 mediated ER stress or other suppressive pathways, as long as the target is tumor specific. And I think that's another difficulty, right? Because there are very few things that are accessible outside the tumor cell that can be targeted without killing the host, right? And I think part of the success of CAR T cells against hematological malignancies was to hit the jackpot of finding CD19, which is a target that, you eliminate the healthy B cells, but you can live without healthy B cells, right? But you cannot live if you target something that is expressed in the lung or the pericardium or wherever, and my concern is that some of the clinical trials are targeting some of these molecules.
It's going to come down to Sohrab helping us, right? Because
exactly. Yeah, totally.
Going to have to show us which are the mutated antigens that are presented. So if we know what these neoantigens are and what these mutated antigens are, maybe it's about and hopefully they span more than one patient's tumor. Then it's about generating the CAR T cells, or maybe even using the endogenous T cells that recognize these neoantigens, but finding a way to expand these endogenous T cells, so that now you can take them out of the body, expand them, and then give them back to patients at, a lot more than they have in there. So even if they're at low frequency, you give them at a much higher frequency so that they can have their effect. But their effect is now more against these mutated antigens that, get expressed on MHC1 or MHC2. And it's not just about the ones that are on the surface, where you're generating these high specific TCRs that can then see any small amount of the antigen, right?
Actually.
I think, one of the frontiers, we just actually hosted an amazing talk by Kathy Wu today. And she was talking about the different forms of mutations that accrue and their ability to create neoantigens. And So for example, what I'm talking about are structural variations and inversions and insertions and deletions. And we're only, and all of the modeling work that's gone into point mutation is really from viruses and things like that. So, we're far away from being able to predict computationally what a mutation, how a mutation would actually elicit a recognizable feature to the immune system. And so I think there's a long road ahead, but I think the data is accruing where we can start to now maybe model these. And again, I think the type of, this is actually where the model systems can be really extremely helpful, because we can probably generate lots of different forms of mutations and expose them in a model system environment. And that's going to be a very powerful substrate to learn the rules of recognition.
Well, this is a great way of ending with a short discussion about your thoughts on vaccines, right? Particularly now in the context of mRNA vaccines that are easy to produce and apparently very effective. What should we use to vaccinate patients? Should be personalized vaccines? Should we try to identify commonly, common immunogenic targets, cancer testis, antigens, whatever, right? And when and who?
Yeah, well, hopefully they're using this mRNA platform now to start back on the cancer vaccine, space, because initially I think the mRNA, vaccine strategy came out from the cancer immunotherapy thought process that you can use these to generate effector T cell responses. But again, because the immune system is this yin and yang trying to control itself, I think we gave a lot of vaccines in clinical trials thinking, oh, we hit the jackpot because we have an antigen that, like cancer testis antigen, and we have effector T cells in the peripheral blood that we can measure by ELISpot assays. The problem was we weren't thinking what happened to those T cells, a few days later when all these inhibitory pathways are now being expressed to dampen that response. So now that we have immune checkpoint therapy and we understand more about the mechanism of how a T cell response is regulated, I think it's time to come back to the vaccines, but think about them as combination strategies. We should definitely not be exploring vaccine as single agent treatments, right? First of all, we know they're safe. We've given hundreds of vaccine clinical trials, so we don't need another safety study. What we do need are rational combinations so that the vaccines and the appropriate immune checkpoint agent can be given. So I think that's how we need to move forward. I'm hoping that the companies that have taken us through the COVID-19 vaccine with their mRNA platforms will be interested in moving that forward into cancer.
Mm-hmm, mm-hmm. Yeah, I also think that there are some diseases or some clinical settings where we can really exploit, a relatively intact immune system. In the context of ovarian cancer, that moment comes after the initial treatment, because typically, surgical debulking and chemo initially work, and then you get a window of opportunity before the tumor reappears, where probably you can induce an immune response that hopefully will be protective, obviously in combination with immune checkpoint inhibitors, etc. So, well, this was a very exciting discussion. I learned a lot, and I think we are 1 minute over time. So we have a beautiful paper here. Hallmarks of response.
This is Pam's review paper. It's amazing.
Good. Highly recommended.
Fantastic. So I ran to read it right now. Thank you very much.
Great.
Thank you so much.
Thank you, Jose, for leading us in this really, really exciting discussion, and to all of you for your contributions to it. It's really exciting to think about the prospects on the horizon now for cancer therapies. So thank you for all of the work that you're doing on that.